So we've been talking about so far how flux analysis techniques really assume the steady state distribution. They're not usually thinking about how regulatory networks are layered on top of all of this. And you can start to think about how, okay, regulatory networks and regulation in general can affect quite dramatically what your distribution of fluxes are. That's just the first case if you're thinking about organismal physiology. Then as an engineer, you can ask yourself, if you used flux analysis, flux balance analysis in particular, to try to predict what gene to knock out, you first of all might find that there are more genes that you need to knock out that just have to do with regulation rather than mediating the particular fluxes and reactions in the pathway. You might also find that you might have to deal with regulation in other ways and genes more generally in other ways than simple knockouts. And so that's actually what homework 4 is really oriented towards testing, is to give you guys some experience and in thinking about the different ways practically that engineers, and specifically metabolic engineers, have to deal with regulation, not just of genes, but even downstream, and come up with solutions, and then how we can harness those as tools. So this overall unit, as I've designed it, will consist of 3 different sets of slides. The first about what natural strategies are, primarily oriented around gene regulation. Then a unit on expression, or well, not a subunit, a lecture on expression engineering that goes over what are the tools that metabolic engineers have specifically used to design control of pathways. And then taking advantage of what the machinery is for mediating gene expression, gene regulation, we as metabolic engineers can start to do more interventions in pathways or more monitoring of pathways based on our ability, often evolved from just natural components, to sense and respond to pathway metabolites. So I think this is collectively a really interesting area that synthetic biology has made a lot of contributions in. And we've talked about, under this traditional framework, how enzymes are obviously the ones performing these reactions, and we've focused on some protein engineering, but we're really going to back up now into a lot of the gene control, effectively how much of the enzyme do we want to make, when do we want to make it. And again, we've talked about some of the opportunities and also some of the limitations of flux analysis techniques. So here we go into gene regulation and expression, biosensor engineering, and we conclude with this notion of gene circuit design, which is kind of an exciting area conceptually, brings in a lot of process engineering and electrical engineering principles, logic, and computing as well, because effectively a cell and any form of life is really is programmed, with some kind of code to perform some kinds of functions, and to do so through often logical operations. To keep everything relatively simple, we're going to be focused on prokaryotes. And we're not going to get through all these slides today. As I hinted, we'll cover some next time. Okay, so let's just start with a big picture view on what are all the different ways that nature has come up with regulation. And this isn't even going to capture all of them. And there's a lot of regulatory processes that we could talk about how cells differentiate, about how cells sense their environment, even simple things, human behaviorally related to like fight or flight response, neuronally. There's all kinds of regulation of biology. I want to keep it somewhat focused on the idea of metabolism. And so I think this figure from a review published in 2011 does a really nice job of tying together why it is that we care about regulation and how it affects metabolism. So we've got these different regulation mechanisms. I list a broad list here on the left, and I'll go over that in a sec. But let's just take 3 examples: gene expression, post-translational modification, allostery or allosteric regulation. And we know that they can affect your enzyme capacity. We're talking about enzyme capacity here as both, you know, what is the actual level or concentration of enzymes that we have, and then what are its kinetic parameters. Because, for example, if you have, say, an allosteric inhibition, you haven't changed the number of enzymes that are present in the cell, but you've affected their kinetic parameters. And maybe not through the example that I just gave, probably wouldn't have this Michaelis-Menten form exactly. You might have another inhibitor species, and this may be dependent on its concentration. But you can see how this idea of capacity is lumping together both activity, generally speaking, affinity, and concentration. So you can have metabolic feedback from different metabolites performing or mediating, controlling those kind of responses. You can have the environment affecting both the concentrations of what metabolites you have, as well as molecular cues in the environment, be they quorum sensing more generally for how many cells are around, a specific cofactor or metal that cells require for certain reactions or respond harshly to stress. And you see that affect your metabolic network and mediate some of these. And then, of course, how active the enzyme may be actually also affects the flux distribution. So you've got all these, this complex interplay here. So thinking about the different kinds of regulatory modalities, I know I said we would focus mostly on prokaryotes, but I thought it's important to highlight, and there are a few examples in prokaryotes, although predominantly this is a eukaryotic phenomenon. This idea of even before you transcribe a gene, when you're just looking at, you know, genes, the DNA sequence alone, there can be epigenetic changes, say DNA methylation. This is really big in, say, human cells. In our bodies, for example, there's a whole layer of pre-transcriptional regulation, chromatin, and how whether DNA is even accessible to transcription machinery, epigenetic silencing or marking. A lot of these processes are what allow all of our cells effectively to have the same genome, but to be, you know, different. I'm, again, not going to go into the details of differentiation, but ultimately your liver cells or your neuronal cells or your skin cells have the same genome, but they have completely different genes that are being turned on. And so that's also mediated directly at the transcriptional level by sigma factors, repressors, activators, mostly a eukaryotic phenomenon, or enhancers. So some of these are DNA sequences. More of these are proteins that bind to DNA. And we've talked, I think, a little bit in the beginning of the course about just the basics of transcription. This lecture of the three that I'm going to give in this unit is the most about genetics and biochemistry, just generally what the cell does. But I'm going to be trying to pitch everything in a kind of engineering way. And one of the things to realize about all these different layers is that almost like within a chemical facility or processing plant, you can imagine the one main control room where you've got like maybe the big red button that says turn off everything, and like the big knobs that really shift your flow rates through major unit operations. But then maybe thinking about a different layer for lockout or tag out, or even just for any kind of measurement or small modulation of a specific process, you might have an actuator or a valve that's right there, located on the unit, that does something small. And so similarly, nature converged around this idea. There's reasons why you have all these different regulatory modalities, and they create really interesting effects. Again, maybe some of you have taken process control. There's a lot of parallels. So once you've made RNA, it's not necessarily guaranteed to give you protein. You could have RNA interference, and this is a tool that's been used by engineers to really dynamically, for example, shut down. You haven't shut down the making of RNA, but now you've shut down the translation of it. Secondary structure, you can just build into the RNA a self-regulating kind of switch. It's harder for it sometimes to be a switch because secondary structure kind of implies here that the RNA, as it's made, has folded up on itself, thus affecting its translation. The difference with something like RNA interference is that you could have another RNA that's complementary and antisense, say, and then it just spines, and maybe you can control the concentration of that. So you have different levels of ability to adjust some of these. A ribonuclease site is just a site that you can build into your RNA sequence for a nuclease to cut. And then in eukaryotes, you have splicing and introns, which can allow you to have, commonly even 4 different isoforms, 4 different proteins made from the same RNA sequence. So that's really cool. Okay. Translational, we're going to be spending a lot of time, particularly next lecture, talking about ribosome binding sites. And even once you've made an enzyme or a protein, we just talked about allostery and inhibition, but there are other things that could happen to your enzyme. It could be post-translationally modified in a way that now has changed its conformation somewhat permanently, or has changed binding affinity temporarily. We tend to assume that once a protein is made, it kind of just hangs around, and certainly the half-life of protein is usually on the order of hours, so it is a lot more stable than, say, RNA, which is a half-life on the order of minutes. But you can actually make a protein sequence, and nature has done this too, have a half-life of just minutes by attaching or appending or just revealing certain sequences that induce degradation rapidly. And part of the reason for this is when you think about the cell, if proteins lasted a really long time, you would have a really difficult time trying to adjust to a new environment. I know I've touched on some of these topics before, so hopefully that was a review of what we had covered a while ago, but in more detail. We're going to spend a lot of time today on transcription.